I hope this session will be informative, and I'm planning to take as many questions as possible. So it doesn't have to be mostly lecture; it can be mostly Q&A if you prefer that. You'd have to be pretty much in a coma, or to have been in a coma for the last eight or 9 months, not to know about coronavirus or COVID-19. And so I'm just going to very quickly give you some details about the virus, give you a more up-to-date sense of where we are than some of these slides might portray, because I didn't have a chance to update them today. I apologize. And then we'll talk about where we are with developing treatments and vaccines, and perhaps what the future may hold in store. So very quickly, I think many of you know that this disease appeared near Wuhan, China. There's reasonably good evidence now that the first cases probably occurred in people in early December or perhaps in November. And this represents the classic example of a virus making the jump, if you will, from an animal species into people, and then unfortunately being able to be readily propagated from one person to another. There are lots of viruses that in animals that can infect you and make you sick. Rabies is a good example, but with very rare exceptions, people don't transmit most of those viruses to each other. So we don't end up having human pandemics. But when one of those viruses, which is completely new to humans, is able to cross over from animals to people and be readily transmitted, we can have the kind of situation we have with COVID-19. So COVID-19 is caused by a novel coronavirus, SARS-CoV-2, and this tells you a little bit what type of virus it is. Lots of genomes have been sequenced, so lots of people around the world are busy sequencing the virus. There's relatively little genetic diversity so far, and very little evidence that the virus is evolving or changing quickly. And that's at least good news, because the hope is when we have vaccines, the virus will not quickly evolve in a way that means the vaccine won't protect the way influenza virus does. I think there's universal agreement by people who are not politically motivated that this virus originated either in a bat or perhaps in a pangolin. You can see what a pangolin looks like here. There's absolutely no scientific evidence that this represents germ warfare or laboratory manipulation of a virus the way some have alleged. This is almost certainly a naturally occurring virus in animals. Exactly how it got into people remains a source of some dispute, but it clearly did. And now we have this very large global problem. So again, by all reports and by molecular clock studies, the virus probably first got into people in the middle of November, and may very likely represent a transmission from a single infected animal into humans. Oops, trouble with that. I won't use that anymore. For those who care, other coronaviruses, corona is the Latin word for crown. When you look at these under electron microscopy, they have little crown-like projections. And other coronaviruses do cause disease in people. Many of the common colds that you and I get every winter are caused by the 4 coronaviruses listed here. They're self-limited. They never make people seriously ill or kill people. In 2002, you may have heard of the SARS epidemic, and SARS was caused by a novel coronavirus at the time, also originating in China. And MERS, Middle East Respiratory Syndrome, is also caused by coronavirus, and that's primarily a virus that occurs in camels and then in people who are exposed to camels in parts of the Middle East. This is, I think I'll skip over this. This is SARS. This is to say that the COVID-19, as of back in August, the global toll, and this is of course a substantial underestimate, was that there were about 23 million people had been infected, documented to have been infected, over 800,000 deaths. According to today's numbers, It's more like 43 million people have been infected worldwide, and over 1.1 million deaths have resulted. But these are underestimates. We know that far more infections have occurred, and we also know that there are many, many more deaths than have been counted in these numbers. And I'd be happy to talk about why it is we have trouble counting the numbers of cases and the numbers of deaths reliably. This is only slightly out of date. This just gives you a global sense. Many of you are familiar with the fact that the pandemic has been particularly bad in some European countries, Italy notably early on. The United States, unfortunately, is a global leader in this, but Brazil and Colombia have been very badly affected as well in South America. And there are now very, very large numbers of cases occurring in India. But of course, the pandemic began in China, and the initial very, very large cluster was there. But what this map doesn't show you is all the cases that really have not been confirmed, and pretty much the virus is present globally at this point and causing a great deal of problems around the world, including on the Berkeley campus. I first gave a talk on this disease on February 6th, shortly after the announcement about the disease reaching the United States. And you can see that on February 6th, obviously 10 months ago or so, we knew of 12 confirmed cases in the United States, 6 in California. And then by August, as I said, we had close to 6 million and 176,000 deaths. And now the numbers are much, much greater than that as a result of substantial ongoing transmission. So anybody who thought this disease was going to just disappear in the summer when the warmer weather came was delusional, and that prediction certainly did not bear out. And frankly, I thought it was an unlikely prediction in the first place. This is a map, again, a little out of date, but showing you that while initially very large outbreaks occurred, you're familiar perhaps with the one in Seattle. There were large outbreaks in Southern California and Los Angeles, very large outbreaks in New York and New Jersey. But then over the summer, the infections appeared in large numbers throughout the southeastern United States, very large outbreaks in Florida, in New Orleans, in Mississippi, in North Carolina. And if I showed you the map as of today, you would note that now a huge part of the problem is in the upper Midwest, in states like North Dakota and Idaho. So the virus basically may have taken its time getting to some parts of the United States or the world, but it is now ubiquitous and present everywhere. And that's a problem. This is just to give you some sense of how the extent to which we underestimate the number of infections. This is looking at estimates back when there were 23 confirmed cases of coronavirus in the United States. The estimated numbers of infections that had actually occurred, just 5 cities: Boston, Seattle, Chicago, San Francisco, and New York. So while we knew about 23 confirmed cases, you can quickly do the math here and see that there were estimated to be over 30,000, or about 30,000 infected individuals by that time. So again, we have trouble really knowing what the total numbers are. Just to show you, there have been studies here in California and elsewhere that pretty much document wide-scale infection of people who either develop no symptoms at all or who are mildly ill and don't seek care. So it's possible that in the most heavily affected parts of the United States, somewhere between 5 and 10% of the population has now been infected, although many have not had any illnesses. And that, of course, raises the question or points out the fact that many people can have this infection and get better. They may develop immunity and be immune to future reinfection, although that's pretty much unknown at the moment. But of course, the fact that many people get infected and don't get sick has raised a substantial controversy about how we should try and control and prevent infections, with some people arguing that because it's mostly elderly, infirm people, the kind of people who live in nursing homes, who are most at risk of getting really sick and dying, that perhaps we should let the virus circulate freely in the population, make everybody immune, and try and circle the wagons, if you will, or protect the frail elderly, but not interfere with day-to-day activities of the rest of society because of the enormous costs economically and socially and other ways that all the various public health measures have on the population. And I'd be happy to talk about that vibrant discussion. Perhaps disagreement might be a better way to put it. So again, these infections are often asymptomatic. Some estimates are that 40% of infections with this virus may be asymptomatic. Other people have a mild self-limited respiratory illness with perhaps a little fever. But unfortunately, some proportion of people develop life-threatening infections of the lung. And the case fatality proportion is something we can talk about, but it's clearly vastly higher in the elderly, particularly people over the age of 75 or 80, and vanishingly rare, but not zero, in young children. So it really does depend on how old you are, what your risk of dying is if you get this. You know, how are we doing in terms of treatment? Well, our treatment has gotten a little better in terms of what I'd call non-specific supportive care. Of the things that were listed here a while ago, I can tell you that the evidence is that chloroquine, despite being promoted by some politicians, is of no known benefit and some potential harm. There's an article in the New York Times this evening saying that their study of a monoclonal antibody shows that it does not work in hospitalized sick patients. There's still some hope that it might be effective at an earlier stage of the infection, but a trial of monoclonal antibodies done by one drug company was just halted today for what's called futility. Some evidence that an antiviral drug called remdesivir can shorten the course of illness if given early. So there are some good things on the horizon with regard to treatment, but there is no magical cure. There's no magic bullet. Just a couple of quick things here. For most people, the incubation period is in the range of 4 to 7 days, but in some it can be shorter or longer. You've heard a lot about routes of transmission. The virus is clearly transmitted through the droplets and aerosols that you create when you cough or sneeze, and probably almost certainly even with loud talking or singing to people who are in a nearby environment. This is particularly a problem indoors in poorly ventilated spaces. And on average, in a virgin population, one infected person would give rise to about 1.5 to 4 new infections. And to give you some perspective, for influenza, it's about 1.5. For a measles virus, it's about 20. So this virus is highly transmissible, readily transmitted from one person to another, particularly in crowded indoor circumstances. I think I'll skip over that. So what are the various prevention control methods that we're using? Well, we isolate cases in the healthcare setting and give protective gear to healthcare workers. Again, there's an article from the CDC in today's newspaper pointing out that a substantial proportion of cases in recent months, unfortunately, have still been in healthcare workers, and that nurses are the highest risk group as far as we can tell. But doctors and respiratory therapists and frankly, other hospital staff have certainly gotten infected. So we're doing our best to protect them, but it's by no means perfect. Quarantine of exposed individuals, we can talk about. Contact tracing, which you've heard about, is meant to allow us to identify people who've been exposed and to quarantine them before they can infect others. This is a very challenging thing to do. I'll skip over the cordon sanitaire, but masks and face coverings, as you're undoubtedly aware, are considered controversial by some people. All of the evidence is that they're at least partially and actually pretty effective at reducing transmission of this virus, but they become highly politicized, and unfortunately many people consider wearing a mask or a face covering a sign of political views or timidity or something. So that's quite a problem getting people in some areas to wear masks and face coverings. There clearly have been travel restrictions and border closings, lots of social distancing resulting in closed schools, canceled sporting events, and the like. Hand washing is a good public health measure. It's not clear how well it prevents transmission of this virus. And then some other things I won't bother to get into unless you have questions about.
Dr. Reingold,
Yes.
we have a
please.
question in the chat.
Great. Okay.
This question is from Sammy. Why would a virus be lethal given they depend on their host for survival? Wouldn't there be evolutionary pressure against that?
Well, that's a great question, and I'm not an evolutionary virologist, so I'm probably going to butcher this. But you're right. If an agent kills everything it touches or infects, that might prevent it from surviving long. But as I pointed out, a substantial proportion of people get infected are asymptomatic or have a mild illness. So the virus from the beginning has known enough not to kill everybody, not to anthropomorphize it, but if the virus ends up killing some people and not killing others, it looks like it can do quite well for itself. And why some people die or get very sick is more a function of how ill the host is in the first place and perhaps an overzealous immune response. So I don't think this virus is going to have any trouble sticking around, despite the fact that it kills some proportion of the people it infects, which many viruses do. I mean, there are viruses like the measles virus and influenza virus that have been around for a very long time, as far as we can tell. And they kill some of the people who get it, depending on host factors. So I don't think that's that unusual. I hope that answers the question. Are there any other questions in the chat? Okay, so what I'd like to do is actually switch from all of this stuff and talk about vaccines and where we are with developing vaccines, so I can allow lots of time for questions. And also perhaps I can talk about the Berkeley Safe Campus Initiative. Maybe some of you were participants in that study that we did this summer, but if not, I'd be happy to talk about that and what we learned. So I'm going to switch and I'll post both sets of slides. But is this showing? You have the vaccine slide up?
I don't see anything right now.
Okay, then I've screwed up, so hold on a second. How do I get out of here? Sorry, I'm pretty incompetent at this.
No worries.
How do I get out of here? And show. Okay, get rid of that. Share screen. Nope, now where did that presentation go? All right, just a second, sorry. Let's see if this works.
Yeah, we can see it now.
Okay, that looks okay.
Before you start, there are a couple more questions in the
Sure.
chat, if you don't mind. So we have one from Noah Parker. Do you think it would become less lethal over time?
So, people are worried about it becoming more lethal over time. People have raised speculation about it becoming less lethal over time. What I would tell you is that for all of the coronaviruses that we know about, they evolve incredibly little over time, as far as we know. So there are viruses like the HIV, cause of AIDS, hepatitis C, influenza. There are viruses that evolve incredibly quickly. And in fact, in some cases, you have multiple strains within you just because it's evolving within one infected person. But but other viruses like the measles virus and these coronaviruses do not appear to mutate much. And so I would not expect, based on what we know, to see it evolve very much in either direction. I could be wrong, but that's my guess. And any other questions about?
There is one more from Jacob. Could you touch on the pros and cons of wastewater testing versus the testing of individuals?
Great question. We're doing both on the Berkeley campus. My colleague, Cara Nelson in engineering is setting up wastewater testing. So for those who are not familiar with this issue, individual testing, of course, is when I come and stick a swab in your nose or your mouth, or unfortunately your nasopharynx, which is quite uncomfortable, or have you spit into a tube and I test to see whether you are currently infected or not, right? That has some advantages, but it's expensive and unwieldy and, slow turnaround times, other problems I'd be happy to talk about. But the Berkeley campus and many universities and businesses are doing that. Wastewater testing, the virus in a human with COVID-19 is present in the feces, in the stool. And so the notion is that if I test sewage, I can detect the virus in the sewage, and that would tell me that someone upstream of where I sampled it, who pooped into the sewage, was infected with the virus. And that we can potentially do surveillance, if you will, on different parts of a city, or dormitories, or buildings, or fraternities, by testing the effluent, the sewage coming out. And if we get a positive in the sewage, that tells us that someone in that, that fecal stream, if you will, upstream, is shedding the virus, and we could then potentially go and sample all the people in that building or dormitory or whatever. That's in its early stages. And how helpful that will be in the long term, I think, remains to be determined. It turns out that it's not so easy to sample sewage. The environmental health people need to agree, and getting your equipment down into sewers is difficult. And so people on campus have been working closely with the city of Berkeley and collaborating with people at Stanford on the science. So in theory, it can work. In practice, it's a work in progress. Let me put it that way. Does that help? Okay, so we have this virus, which is now ubiquitous pretty much around the world. And if you read the newspapers in the last few days, I know young people don't read newspapers anymore, but some source of news, you will know that we've had substantial increase in the numbers of cases here in the United States. Some of that is due to more testing, but some of it is due to spread of the virus into rural areas, into areas that weren't that affected a few months ago. Some of it is related, I'm sorry to say, to college students going to parties and other events and transmitting the virus readily and then into their communities. So some of it has to do with people refusing to wear masks and going to bars and other settings. But the fact is, we're having a resurgence of the virus here, as well as in Europe and in a number of other countries that at least initially thought they'd controlled things. And really, a lot of hope is being pinned on a vaccine. So let me quickly talk about vaccines a little bit in the next 5 or 10 minutes, and then we'll have about 15 minutes for questions. So we'll skip over all that. There are now, I think, close to 200 vaccines under development in different countries and by different companies in various stages. Some of them are going from the bottom up, the whole virus, where you basically grow up the virus and inactivate it. So it's an activated whole virus vaccine. Some are vaccines where you take one or more proteins from the virus and purify and reproduce the proteins, so the virus itself is not present. Some use what's called a viral vector, typically an adenovirus or another virus that doesn't harm people, but it's a live virus vector. You engineer it to carry one or more of the proteins of the coronavirus to induce immunity to those proteins. That was used successfully for Ebola vaccines a few years ago. And some are completely modern approaches that have never been used before, using mRNA to basically deliver the code for one or more viral proteins into human cells in the body, which will then basically make these viral proteins, and the human will make an immune response. No mRNA vaccine has ever been licensed or used in humans before. So some people consider them quite experimental, but we've got many people doing many different things to try and come up with a coronavirus vaccine. This is out of date. I can tell you that as of the New York Times coronavirus vaccine tracker today, there were 34 vaccines undergoing safety and dosing testing in the United States. There were still 14 in what are called phase 2 trials. There were 11 in phase 3 trials. And there were 6 vaccines approved for early or limited use, but this is not in the United States. This is in countries like Russia and China, which have basically decided by testing a vaccine in a couple of dozen or 100 people, they can determine that it's immunogenic and safe and start giving it to large numbers of people. But so far in the United States, no coronavirus vaccine has been approved for use. And there's been a lot of politics around when we will begin to get data from those now from the trials that are currently happening. Whether it'll be by election day or not, I think it's pretty unlikely to be, but I wouldn't know for sure what the president is planning. So many different vaccines in different stages of testing. Just to quickly explain that preclinical testing of a vaccine occurs in animals, mice or monkeys, to see if it makes an immune response. Phase 1 studies are in people, typically a few dozen or maybe 100 people or something like that, typically healthy adults like you guys. And it's to basically judge safety, look at different doses, and confirm that it stimulates the immune system, makes antibodies or cell-mediated immunity. Phase 2 studies basically typically involve hundreds of people, and they test different doses, look again at safety, stimulation of the immune system, and typically are then done in the age groups or the target groups you want to target for vaccination. But only phase 3 studies test the efficacy of the vaccine to prevent the infection or the disease itself. And these typically involve thousands of people. In the case of the COVID vaccine trials in the United States, they're typically in the range of around 30 to 50,000 people in each of those trials. And you basically randomize half the people to get this new vaccine or to get a placebo, and then you follow them over time to see who gets infected or gets sick and who doesn't. And you compare the rates in the vaccinated and the unvaccinated to see how well the vaccine works. Some people have suggested, and the British are about to embark on what are called challenge studies, where you actually intentionally infect individuals with the virus as a way of getting efficacy data more quickly. But the challenge studies will not be done, as far as I know, in the United States. So as I've said, Russia has already approved a coronavirus vaccine, although it has not proven to be, there are no studies showing it's efficacious, and safety data are not really available in large numbers of people. China has done the same thing. And just to say that when we're testing a new vaccine, we want to know whether it prevents the disease or not. If it doesn't prevent the disease, it doesn't matter if it makes an immune response. We're trying to prevent the disease. Some vaccines will prevent transmission of the agent to others. We, of course, really want to know how well it works in different subgroups. Does it work well in children? Does it work in old people like me? Does it work only in healthy people like you guys? And of course, it's vital to know about the short-term and the longer-term safety. And often we want to know what happens if you give it at the same time you give another vaccine, such as influenza or some other vaccine. So there are many, many questions to answer about these vaccines. And we really won't have answers to some of these questions for a while. And I will point out that there are other viruses, bacteria, and parasites that where people have been working on vaccines for decades, for which we still do not have safe and effective vaccines. So it is by no means a certainty that we will have safe and effective coronavirus vaccines for COVID-19. But I think the people who know most about it are optimistic that we will in fact have them potentially within the next several months. We've occasionally had what I call safety surprises, vaccines that were thought to be safe and effective in the trials that were done. But when they were given to much, much larger numbers of people, millions or tens of millions of people, turned out in rare instances to cause some pretty severe adverse events, maybe in 1 in a million people or 1 in 200,000 people, which we couldn't have known from the trials that were done. So we're obliged to study vaccines even once we've licensed them and giving them to large numbers of people. So as I said, there are a number of the vaccines that have gone through trials. The other issue, of course, is once you've shown a vaccine is safe and effective, you need large numbers of doses. So you need manufacturing, you need to put it into the right kind of containers. You need those to manufacture those containers. You need to distribute the vaccines. You need to track who's getting them. You need to decide who should be in the highest priority group to get them first. You need to worry about equity in our highly unequal society in terms of healthcare access. We need to monitor, as I've said, effectiveness and safety. And we need to worry about all those people who say they won't take the vaccine even once it's available. And just to say that of the coronavirus vaccines that are being tested, most of them require a 2-dose schedule. So if we give you 1 dose, we need to track you and make sure you get a 2nd dose of the same vaccine at the right time, and you need to be willing to get the 2nd dose. Unfortunately, at least 1 or 2 of these vaccines in advanced stages has to be stored on dry ice at -70 centigrade throughout its transit from manufacturer to patient. And so that imposes some real logistical challenges. The necessary cold chain, most of our vaccines do not require storage on dry ice to remain potent. And at the moment, about 40% of the US population says they won't take the vaccine even once it's available because they're worried about its safety. They're worried about the quickness, the speed with which it's been developed and tested, and they do not want to be at the front of the line. So we have a lot of work to do, not only to get people to accept the vaccine, but to make sure it equitably reaches hard to reach groups and convince them to take it. And we have all these other challenges. And most estimates are that even under ideal circumstances, we won't have enough vaccine for everyone in the United States who wants it until at least the spring of next year and possibly the summer. So a vaccine is not going to ride to our rescue in the next few months, whatever you may have heard from political leaders. And I think I'll skip that, and we'll take about 15 minutes of questions.